Everyone. In this video, this is going to be something mostly for the graduate students. For the undergrads, if you want to watch this video, that's totally fine with me. Part of what we're going to be talking about here is something called, or are some issues that come up when you start analyzing real world data. Part of this is going to be how do you actually design an experiment. Part of it's actually going to be how do you analyze that particular experiment. And we will see as we're going through how sometimes these things can actually get to be fairly complicated. Some of the ways we're going to analyze the data in here are actually based on things that you already know. Some of the things we talk about in here are going to be brand new concepts. None of this is really going to be on an exam. None of this is going to be something that I'm going to test you on in the lab or anything like that. This is just things that are really good to know so that when it does come up, and these types of things do come up fairly often, you'll be able to see what's going on and have an idea about how that's actually going to work. And uh, everything we're going to be talking about here is actually material based upon a conversation I had with a researcher. He's an educational researcher who's working in the or with autistic students and specifically autistic children, like ages say 4 to 9 years old, something like that. One of the goals is to teach the children lessons, and then another goal is to see which one of the methods that they're looking at actually works the best. And they ended up doing a pilot study instead of with the children, with the full grown adults. So there's a lot that we can actually look at here. Um, the analyses can get to be a bit tricky. Part of the reason for that is, despite what a lot of the statisticians want to tell you, uh, just basically uh, some of the terminology that we use is not standardized. It's not well understood by everyone. Not everyone uses the same definitions and same everything, which makes the what I'm about to do here a whole lot harder. Also, when you look at, say, how you'd analyze these data in SPSS, that makes it really tricky too. So I'm going to try to give you everything that I can based on what you might find if, say, you're reading, say, a psychology article or versus, say, a biostatistical article that could actually be on the exact same thing. There'll be some terms that come up in both places that are totally different. If you happen to work with someone that's into what's called econometrics, so statistical modeling of economic data, they're going to have some of their own terminology for this as well. And if you're already going, oh, dear God, that is the correct thing to do. I'm not even going to lie about it. Part of the fun is when I'm, because I've had a bunch of these different types of classes, when I'm standing or sitting around listening to status or people that use statistics talk with other people that use statistics, they can be talking about the same things, but one group has a certain definition for it, another group has a different definition for it, and then those two definitions for that word don't always match up. So they're looking at each other like, do you really know what you're talking about? At least that's what they're thinking. So I'm going to go through a couple of these different cases just so that you can see the types of things that are going on here. One of these is fairly basic. One of these things is a little bit more involved. So I will start off with the basic one first. And it does mean that I have to flip the camera down. And it means that I have to flip or rotate the camera as well. So bear with me. So what's going on here is they had a bunch of adults come in, and what they're looking at is an intervention A with two different levels, and we'll call it intervention B with two different levels. Intervention A is a bunch of people are literally sitting in a room, either talking to and listening to a robot while being recorded. The other group is actually listening to a person tell essentially those same stories. Intervention B is dealing with the stories, and the stories would be sad or surprising versus, say, happy stories. What do they mean by sad stories? What do they mean by happy stories? I didn't listen to the stories, so I honestly don't know. And the way that they had this up is, let's say they've got 10 volunteers. And what's going to happen is, say, volunteer number one is going to be assigned to, say, the robot. Then they're going to hear both the sad and the happy stories. Volunteer two will listen to the person, and they will hear the sad and happy stories. Then, say, volunteer three, because these things should be randomized, probably have it like, say, person, sad, happy, and all of that. And the response they're looking for is, well, they actually had like a dozen of these. Uh, but like passive methods for showing that you're listening. So like if you nod your head or you say, mm-hmm, something like that, you're not really in deep thought talking with the person. You're just kind of listening to what they have to say. And then there's active responses where you come out and say like, wow, and you make a comment after that, or um, you say something with excitement. So what they've got here is looking at the number of times someone says like, mm-hmm, mm-hmm, yep, mm-hmm, okay, yeah, mm-hmm, versus, wow, that's weird. That sounds interesting. Can you tell me more? Meaning that they're more actively listening and more actively responding. They also had several other responses, which I'm not even going to get into. But what happened with this is they had, uh, I believe it was four numeric responses and two or three responses that were dealing with the percentages. Anytime we're dealing with data that's got percentage, we actually need to go and make a transformation on that. The gentleman who was doing this, I talked with him about this and tried to set him straight on this. With the numeric responses, that's another type of response. So what actually happens if you've got one of these responses where you have, say, a percentage, okay? Uh, if you just have percentages, you can actually come out and potentially use the two-prop z-test. So this is like, say, male, female, yes, no, or in the case that we're dealing with, say, person, robot, happy, sad, or something like that. If we have more than that, we could look at the chi-squared test. Another option, which if you have a lot of variables, and you don't have enough to actually make, say, a chi-squared test work, and because you got many variables, clearly the two-prop test won't work. What you can do is you can take that percentage that you have, turn that into a probability. Once you have that probability, and this is literally how you're supposed to do it, you take the natural log of that probability and you divide it by 1 minus whatever that probability is. What's going to happen when you do this is you're going to come out with a number. That number is going to be continuous, and it can have a range from basically negative infinity to positive infinity. So whatever this number is, you can now use that and turn that into a linear regression, like we talked about a week ago, 2 weeks ago, whatever it happens to be. And this is, if you have this percentage data, again, if you've got group A versus group B, you can use the two-prop Z test. If you have enough data in each group or each cell that you're going to be looking at, you can use a chi-squared test. But just as an example, if we're talking about percentage, we can say, and sometimes we don't even need the percentage, but we need something that we could say turn into a percentage. So we could say score on exam, because you get the score and it's however many points you got divided by the total. That's going to give you the percentage that you need. Then you can come back, take that percentage, use this method right here. So again, percentage, turn it into a probability, and take the natural log of the probability divided by 1 minus that probability. That will get you a number. And from there, we can then go on to say, how much time did you spend studying for the exam? Sorry. Oh, that pencil doesn't seem to want to work anymore. Let's see if I've got something a little bit better here. So you can look at how much time did you study? What was your grade in the previous class? If we're talking about, say, one of my stats classes, highest math class you took, just to see what's going on here. We could even look at, say, number of credits you're taking in a particular semester, which semester it is. Are you taking it online or in person? So we can look at a bunch of these different things to try to get an idea of what actually, or how well you're actually going to do on one of these exams. Since I got the pen here, I'll make this a little bit bolder so that everyone can see it. So number to probability. Then you take the natural log of that probability divided by 1 minus that probability, which is going to get you a numeric value. And then you use whatever that numeric value is. And you can see what is the effect of all these different things that you want to look at. Something that you could do, especially if we're talking about certain math classes or professors and stuff like that. So if we're looking at, say, I don't know, 6 different professors that all teach the same thing, we could look at professor. We could even say, look at the gender of the student, potentially the race of the student, to see, do any of these things have an impact on, say, the number of points that you get out? And just so we're clear, we can also do things where we can look at, say, the professor and the race as a combination. And that can actually help tell you if, say, there happens to be a bias on one of the professors against or for people because of, say, their race or because of their gender or anything else. Does that mean that, say, the professor is racist or the professor is sexist, depending on whatever? No, it's just giving an indication that there's potential for a bias there. Then you have to look and see how big that bias is. And just so we're clear, when I'm looking at all of this stuff, for me, all the different races that, or at least the data that I have for all the different races, everyone's about the same, but there is like a 2 to 3% gap between some of these. Gender, same thing. So, well, actually, gender in some math classes, the women just do a whole lot better than the men. In the gender in other classes, the men and the women are statistically equivalent, meaning there's no significant difference between them. So a lot of things that could actually be going on here. All sorts of things we can think about, talk about, and deal with. And then for the other part of this problem, they've got, I'm going to call it outcome number 1, outcome number 2, outcome number 3, outcome number 4, versus intervention 1. So the robot versus the person. And intervention number 2, sad story versus happy story. Okay. Because we've got 4 different outputs here. This is actually something that we would call MANOVA. So this is just like ANOVA that we covered before, but instead of having multiple groups we're looking at as the input, the independent variables, we have multiple output variables. And the M just stands literally for many. Okay. So because we've got all these outputs that could actually be related to each other and could be related to the inputs, the way that you analyze this is when you go into SPSS, you're going to be looking at ANOVA. And they, depending on what software you use, these are the things that you actually want to test. You want to see if you can, say, come up with a model to help predict these, or come up with a model to tell, is one of these combinations of things better or more engaging, or all the different things that they're looking at here, than the other. And with the input variables that we have here, because these are the 2 things we can control, we would actually come back and call these fixed effects. So what does that mean? Well, fixed effects are things that you can actually control. We decide whether that person is talking with the robot or whether that person is talking with the person. And we can tell or have them listen to the sad story as well as the happy story. Okay. So this gets to be a little bit weird, but there's a lot more information that we can have on the participants than what we've got right here, right? So this is just saying a particular person sits down and either talks with the robot or talks with the person and then listens to the sad and the happy story. What are some other factors that might actually be important here? And keep in mind that these are all adults who do not have or not on the autism spectrum. So these are like your average adults that just came in to participate in this particular experiment. So other things that might actually be important. So we've got intervention 1 and intervention 2. But we might want to know something about race, gender, age, and because all of this was done over a long period of time, say even the temperature in the room when the person is doing this. And I can say, well, what does the temperature have to do with anything? Well, how many of you have ever sat in a really hot or a really cold classroom and tried to pay attention really well to what the professor was saying? If it's too hot, it gets to be difficult and you lose interest really quickly. If it's too cold, you spend all your time thinking, what? I'm cold, as opposed to, I should be learning this, right? Age, gender, race, these are all other things that we can record about the people, but we may not have any control over it. Meaning we can't say, we won't say person 1 to be of this race, this gender, within this age category, and then we're going to give them these interventions. Because when it comes to having volunteers, you basically get what you can get because we have no control over any of these things. If you take certain stats classes, they're going to call these covariates. And up here, the things that you do have control over, they might call them factors. Of all factors are fixed effects, but not all fixed effects are factors. So I think this is actually a bad term to use for the independent variables or the input variables or the predictor variables, whatever you want to call them. But under very specific conditions, factors are the fixed effects, which you do have control over and so on. And now because you have what's called fixed effects and these covariates, or we could call them random effects. We now have fixed effects and random effects. And that will give you what's called a mixed model. So here's where it gets to be even more. And again, I know this is stuff that you've never heard before. And again, I'm not testing you on this. This is just so that you know what's going on and how to do it. If all we have are, say, 4 outcomes and 2 inputs that we control, that would be MANOVA. So if we have, say, O1, O2, O3, O4 versus I1 and I2, that's called MANOVA. On the other hand, if we're going to have I1 versus I2, or say O1, O1, O2, O3, O4 versus I1, I2, then say race, gender, age, temp, etc. All of this together is something called MANCOVA. The C stands for the covariates, which means there are these random effects that you can't really control. You have no ability to deal with them, but they might actually be kind of important for what's going on here. And that already seems like a lot, doesn't it? Now, one of the places where this sort of thing actually happened, if you happen to be walking around the health science or the health HHB uh, at Oakland University, on the first floor when you get into the Wayback Laboratory, you'll actually see that someone actually did 8 or 9 different outcomes versus one input. They should have used MANOVA depending on what their input was. But the other thing that they could have done is, since all they were doing is looking at the correlation between this and this, then that and that and that and so on, they could have made the variable that they considered to be the independent variable the dependent variable and used a multiple linear regression to do the exact same thing, and they would have done a much better job had they used MANOVA or had they used a multiple linear regression and just flip things around. So with this other thing that came up, there were intervention, again, with these kids, robot versus human. And then there's an intervention, lessons. So lesson 1, lesson 2, all the way out to 10 different lessons. So we can think of these as weeks. What happens here is we have child number 1. They get, I'll write this down a little bit further, sorry. Child number 1, they get lesson 1 from both the robot and the human. Lesson 2, robot, human. Lesson 3, robot, human, and so on. Child 2 gets the same thing, robot versus human, robot versus human, robot versus human. And they do that all the way down. And then their outcomes here are basically number of signs students or the kids are bored. There's also looking at number of lessons that they learned, meaning they were somehow tested on it later and trying to see who did the best with all of this sort of stuff. Okay. So here we've got, they started off with 10 children that were supposed to go through 10 lessons. To make it even worse for them, they had a couple children, let's say child number 9. They were there for lesson 1, 2, 3, 4, but then they left. So there's information on what happened the first time they did it. And again, robot and human, robot, human, robot, human, robot, human, however they put it together. But after the while, say the child got sick and wasn't able to come in for a couple of weeks. So they were asked not to come back because that would according to them, say ruin the study. So how do we handle it when you have someone, it just sort of disappears like this? This is all called missing data. If you get into biostats program, there's actually an entire class dedicated to this basic idea. That is how important and how complicated this is. And one of the things that came up while dealing with all these sorts of things is, how do we handle that missing data? What do we actually do? Well, one of the possibilities here, because it was a child and we know how they did for the first 4 lessons, but not for any other, there's a lot of potential for what could be considered a correct method for analysis. So, talked before, because we had students, or if we're talking about a cancer trial and people drop out for whatever reason, we can usually use something called survival analysis. With this basic idea here, because we have child 1 taking lesson 1 from both the robot and human all the way through, we know how this child goes all the way through. So if we have it all the way up to lesson 10, we know what happened there. We have a complete data set, but for that child number 9 I was just talking about, we have their data for L1, L2, L3, say L4, and then nothing else. That child's data would be censored. So that's where this survival analysis type of thing comes in. Another option is, because they had complete data, from 8 kids, and they're looking at lesson 1 to lesson 10, some of the things they can do when they go to analyze these things is called a child, a random effect. In this case, the child is a random effect because if you, we're not going to be looking at the same, say, 8 to 10 children over and over and over again. We're going to be looking at many, many children over and over again, which essentially means we don't have any control over the child on their own. We'll call them a random effect. And a special type of a random effect called a block. The intervention. So the story, we do control that. So that's going to be a fixed effect. We have the number of lessons. That is controlled. So we have what's called a fixed effect. And then we have outcome for lesson one, outcome lesson two, outcome lesson three, and so on. So this gets into how do we want to analyze this? Because this is essentially week one, week two, week three, week four, week five, so on, so on, so on. We have the opportunity to use what basically a linear regression on this or with this data because it is continuous, or essentially it's continuous. We can come back, we can look at all this stuff under this block idea here. And since we want to look at all these different outcomes, we can also do something that's called panel data. Panel data in general deals with observation, or when you have many observations, but they're not, say, all done at the same time. The outcome here is kind of like that MANOVA, because we have the random effects here be a MANCOVA. And we could look at child, story, and then lesson number and look at the outcomes. We can look at the panel data or look at this as terms of panel data. So lots of things we can do here. There is no 100% totally correct, totally accurate way to do this. So let's say we have all of this stuff for child one, and we're looking at story and then lesson. We can have it from the robot. We can have it from a human. These are both going to be noted as lesson number one. And then we can look at, say, outcome number one. What are they for this? Lesson number two, what are the outcomes? Robot, human. Lesson number three. And actually, in this case, there'd just be the one outcome. So week one, how did this child react to the robot telling the story on lesson one? There you go. How did the child react to the human listening to the story for lesson one? What is their result? We can do this. We will block on the child there. We'll have another data set for child number two, another data set for child number three, and so on. And this would basically be how this is done. In this particular case, we call that linear regression. Now, if we write this up a little bit different way, so we're talking about child one, and we're looking at their story, robot versus human, and then outcome week one, outcome week two, outcome week three, outcome week four, and so on. This is going to be more like how we do panel data. So quite literally, depending on how we put the data into the software, we can come up with two very different ways of trying to analyze the exact same data. And with this, the panel data, another good option here would again be MANCOVA. So depending on how you want to analyze it, what it is you're actually interested in, you can go in and you can analyze this data a couple different ways. Is that confusing? Yes. That's part of the reason why it took about 3 hours for me to try to explain it to the person, because I was trying to figure out what it is they're doing, what they want to do, what are they trying to go for. And one of the things that came up, and I'm sorry to say this, but one of the things that came up while we're trying to do this, and this is something that happens unfortunately with great frequency, uh, the researchers think they know what they're looking for and they think they know what they're doing. So they think they know how to set up their experiments. So they think they know how to get the right answers, but they don't actually know what those answers are. There's what they want to get before anything happens, and then things happen during the experiment. What do you do with that? So lots of ways that we can do this. But the important thing here is with when we're looking at the child, we want to take the child into account. We want to consider the child to be what I said called is a block. A block is just a way of saying all the data that was gathered under this block. So say all the data that was gathered by this particular child was done so under the same conditions. Um, so just as an example, if you're going to go out and you're trying to figure out what color do you want to paint a house or something like that, and let's say, or if you're going to be painting parts for a car or something like that, I think this one actually works out a little bit better. You need to know, like, say, what is the proper temperature and proper humidity for painting all these parts. If it's too hot out, the parts won't look good. If it's too cold out, the parts won't look good. So there's a certain range. Humidity may or may not have an effect on that. And it may or may not have an effect on, say, the person or on the paint that you're using. But imagine it's 85 degrees with 82% humidity, and you've got to be in a full-fledged Tyvek uh, paint suit in that. You're going to be pretty miserable, right? Your glasses or your goggles are going to be fogging up fairly easily. You can't really do much to get rid of the fog in there. So humidity and temperature might conspire against you. And what you would do is say you find that one on a particular day of the week, you go out and you paint a bunch of parts and you rate all those parts. And you know that it was done with certain type of paint, there's a certain temperature, certain humidity, and then maybe you know the name of the person that did the paint or painted the parts. If you had 3 or 4 people painting the parts all at the same or painting parts in the say their own tents or whatever at the same time, that's fine. But you'd call all the parts made on that particular day a block. Okay. It's weird, it's awkward. But another way that we can think about it, especially if we're looking at something like this, where if you're not familiar with autism, it's basically a spectrum. Some people are lower on the autism spectrum, some people are a lot higher on the autism spectrum. And if we consider each child as a block, we know that all the data that was recorded was under that child's level of autism. So maybe and these are some things that I've actually seen pop up. You look at all the data all by itself and you don't bother to look at, say, the child, or you don't bother to look at these blocks, and there's literally nothing going on there. And it looks like everything you did was for nothing. But let's say you have these 10 children, 4 of them are at one end of the autism spectrum, 3 of them are at the other end, and then 3 of them are kind of in the middle. Okay. Each is going to start at their own place. each is going to end up at their own place. And maybe, and I'm not saying that this has happened with them, but maybe someone that's lower on the autism spectrum responds better to a human. Maybe someone higher on the autism spectrum responds better to the robot because they don't feel like the robot is, say, staring at them or something like that. Maybe it could be the opposite thing. I don't know. Um, Maybe the children that are in the middle of the autism spectrum, it doesn't really matter which way to go. Once we know to look at each child and have some idea of where each child is on the autism spectrum, now we can try to calibrate what we expect to see. And I've done it many times before. All of a sudden, you throw in that one thing that they didn't want to look at because it doesn't matter. It has no purpose, blah, blah. And it's the only thing that really matters, or it's the big thing that matters here, which is one of those other fun things too when you're analyzing data like this, trying to talk with people who have been doing, say, the type of research that you're analyzing for years, if not decades. And I actually did this with a group of people that at the time when I analyzed it, they had a combined like 250 to 260 years experience. And I came in and said, I was drunk and I was able to figure this out, but none of you guys could. And they were all guys and they were extremely pissed. Can't imagine why. But I got the right answer. I used methods and had thoughts that they collectively had not thought of before. And uh, all of them had basically the exact same background. And instead of having a diversity of thought, diversity of way of looking at things, it became an issue of what's called groupthink, where everyone's got the same background. So if one person says it's right and it makes sense to everyone else, well, that must be the way to do it then. Not even close, but they don't know anything better. They don't know what else to do. Therefore, their way is the correct way and everyone else is wrong. And then you find out later that they had a way of doing it, but their way was a way of doing it. And as they found out, not a very good way of doing it. So I will leave it at that for this video. Again, I don't expect you to know or test you on anything that's in here. This is just so that when this comes up, and it will, you have a little bit better idea of what is actually going on here. All right. Look forward to torturing you guys later.